Erin, can I get started right now?
Yes, please.
Okay.
I started the recording, so we are good to go.
Thank you so much. So, hi everyone, and first of all, I want to express my great thank you for everyone for supporting this class, for attending this class. Hopefully, there's something you will feel useful in the past 2 weeks. And let's finish our last session today. Last time we started with some pitfall of single-cell data analysis, and we still have other pitfalls, and we will finish them today, maybe in 3 to 4 p.m. Session. And right now, I want to mention the analysis techniques. Yeah, like last time, in the last week, we mainly introduced the Seurat, which is a R package. Yeah, it's standalone R package. You can download it on your computer. Yeah, you can use it on your computer and play with your own data. Yeah, so this is one kind of technique, computational technique, and the other kind of technique, so-called web servers. Yeah, or web databases. Yeah. So here we introduce a little bit regarding the IRIS-EDA, which is an in-house tool developed here yesterday, and today we're gonna introduce more details of what is IRIS-EDA and what it can do. And Chang Kun, my student, still be here. We will lead everyone here to go through the website together and check the main functionalities there, so that you can know what kind of analysis can be carried out there. Yeah. So first of all, this is the paper published in PLOS Computational Biology. I'm listing the servers here. Yeah, the first one is the web server hosted at OSU. Yeah, and this is an alternative server. Yeah, and this server was hosted in my previous university. So currently, the main entrance of the server is the first one. But just in case the first one encounter any maintaining or some other issues, it's not available, we will use the alternative server. So basically, both of them are available right now. On the other hand, even IRIS-EDA is a web server, but it's developed based on R-shiny, which means if you prefer use this software on your local computer, you do not want to upload your big data to the server, which might be very slow. You can deploy a local version, from here. So this is the beauty of R-shiny package. You build a server based on R, and if you want to use a local one, you can just download this source code and deploy on your own computer. And then when you open the RStudio, you can use it just like Seurat there. And we have some tutorial on YouTube channel. Yeah, you can check if you're interested in. Okay. So a quick summary of why we need to use a web server, other than the standalone packages. So basically, among all these advantages, at least 4, very significant ones. So no coding skills required. Don't worry about any coding background. You just if you can purchase some book from Amazon, then you can do analysis on a web server. It's a user-friendly platform. Standard workflow. You never worry about the reproducibility. Like when you have a software, you can generate results today. Maybe another day you cannot. Due to different parameter, due to different computer environment. Those kind of things always bother the reproducibility. But here, you can always reproduce it, because this pipeline, this workflow is maintained on in a backend server. Yeah, will not affected by you. Yeah, so standard workflow and an immediate interactive view of the data. It's not like you work on your data on your local computer, and after you generate a table, and then you go to another environment to draw some figures and to draw some other visualizations. Now on a web server, you can see those interactive things there. So you will see more examples when we go through the real server. Okay, so basically this is the title and the main distribution of the functionalities. For the input data, we accept the gene expression matrix, which means a row represents a gene, a column represents a sample or cell, and you have a condition matrix. Yeah, you have an annotation file, yeah, for each gene. So those files are acceptable. Yeah, if you have those file ready, then you are ready to go through all the following analysis. Yeah. The analysis are mainly divided by into two categories. The left side is so-called discovery-driven analysis. Yeah, you have your data. Yeah, you want to explore what's useful information or interesting insights hidden in the data. Then you can do PCA, yeah. You can do clustering. You can do byclustering. You can do sample distance matrix calculation. A lot of things there are ready for you to explore, to analyze your data. Yeah. And the other category is so-called differential gene expression analysis. This is basically hypothesis driven. Yeah. For example, you have two groups to be compared. You just directly want to know what's the differential expressed gene. Of course, those statistically significant genes are there between group 1 and group 2. Yeah, between treatment and control. Yeah, so this is a well-defined package, well-defined functionality here. You can do all kinds of differential gene expression analysis using the state-of-the-art tools. And along with a lot of functionality for visualization, like those volcano plot, four-way plot, MA plot, those things are implemented as a function in this package. But if you are using a server, they will automatically generate it. Yeah. Okay. Regarding the the current citation and intention of this server. So it's published last year, 2019, and have 11 citations so far. And we just recently found out the attention score of this server is pretty high. It's top 5% of all those research output. Yeah, in this domain. Yeah. And we have a lot of Twitter and blogs and other attentions. So people are recognizing this server and using this server like in different further development. Yeah, I just list some of the citations here. Yeah, who citing this software. They either for using our server to further data analysis interpretation, like the first NIR paper. Yeah, or they generate other tools, yeah, like this Jubis. Yeah, this is published in Cell System. Yeah, it's interactive notebooks for RNA-seq data analysis. This is revealed in the Bioinformatics paper, how for comparing byclusters and how to correct it. So the byclassing is one of the discovery-driven analysis on this server. So they should use it and cite our paper. The last one, the BMC Bioinformatics paper, Ep.8. Yeah, I know the corresponding author. He is my previous colleague. After our tool, they also developed a similar one, and with more experimental data enriched. So they cite our package. Yeah. And we are actively maintaining this website. Yeah. And received some like very good feedbacks from collaborators, from users. Like the left one is from a professor of Harvard Medical School, and he communicated with us for several times, like showed his very good attitude, yeah, and in using this tool, and found it very, very useful. Yeah, I think this is one of the very good examples. I do believe as a biologist, without sufficient computational training or background, this server will truly help you. Yeah. And on the right part is another email. I do believe this is another. This is a physician, I think. Yeah, he found that it's a perfect match for what he or she wanted to do. Yeah, with his or her RNA-seq data. Yeah, this is from Michigan State University. I just show some good examples here. Okay. This server currently can be accessed by both of the links. I think Chan Kun is here. He can go through the server, with everybody. Chan Kun, are you ready?
Yeah.
Okay, Chan Kun, you can feel free to share your screen and show the functionalities on the server. Yeah, with the group. Okay.
Okay. Thanks, Dr.
Yeah.
Hello? Yeah. Can everyone see my screen right now?
Yep.
Okay. Yeah. So we are at the IRIS-EDA web server. So as Dr. Ma mentioned, the server is currently hosted on both OSU-MC and OSU and SDSU. So if you don't remember the actual URL, you can just simply search from Google. So we have the results here. So just search the IRIS-EDA. So the IRIS-EDA is short for Integrated RNA-seq Data Analysis and Interpretation System. So you can perform the RNA-seq data analysis and the single-cell RNA-seq data analysis. So to use the server, you need to prepare the input data. So which is the count matrix and a condition matrix. So I will be using a single-cell RNA-seq example here. So this is the data I will be using today. This is the single-cell RNA-seq gene expression matrix, so where the genes each row represents gene expression value, and each column represents a cell. And another file we need to prepare is a metadata file, where the first column should be the exact same, the cell names in the count matrix file, and the rest of the column represents the group information. For example, the second column is the cell type information, and the rest are the timing information and condition information. So once we prepare this input file, we can submit this submit the data to the server. And we also have some examples here. If you don't have if you just simply want to try to test the functionality of the server, you can click on these three buttons to just start from the example data set. But since we have the data, we can first select this radio box and just upload the single cell data. And the second option is the data processing. panel where we will here the default value is 10, which means we will remove the count data row sums less than 10 and perform the log normalize for the downstream analysis, and you can also submit. And we have just uploaded the datasets, and we can. This is an overview. You can see the upload file should be just the Excel we just opened, and the other tab we have the concept or summary. Since it's a single cell dataset, so this section may be kind of slower than the normal IRIS-EDA datasets, because single cell usually have a lot of columns, so it may take some, takes a minute to load this panel. So first we have the count data distributions, and this tells the overview of how this the expression data distributed in each cell. So here each bar represents a normalized total counts of that cell, and you can see the so good data usually the this box should be relatively on a single same line. So, and the next one is the count data distributions. Since we are doing the single cell IRIS-EDA data, you can see that most of the data are zeros. You see there's a very high bar in this zero value, and the rest are very small, and that's also what we expect from a single cell IRIS-EDA data.
Yeah, and this is very good evidence showing the sparsity of single cell IRIS-EDA data. Yeah, showing those dropout issues we mentioned, in the previous presentations. Yeah, Tankun, you can go ahead.
And the last one is the total reads. It's similar to the box plot, but we use a bar plot to just plot the total counts of each cell. And these three plots are mostly used for a quality check before you do your analysis. And next, we can go to the Discovery-Driven Analysis tab. So the first tab, we have the correlation heat map shown here, where each row and the x-axis and y-axis are both the cell names, or in IRIS-EDA just the sample names. So you can see the more correlated data tend to the same cell types tend to have a higher correlated coefficients, and in this plot, it's more yellow in this plot, and we can also export this figure into a static plot, and we have the this is the static version of that correlation matrix, and using a different color scale, and we have to more the red, and the sample are more correlated with each other. So next three are the we have the PCA, MDS, and TSNE. These three are dimensional reduction methods. So we are currently at the PCA, and it's short for principal component analysis. And this is a linear reduction method, and we plot this PCA scatter plot here, and we can color these stars by the annotation file we used. We can either color them by the cluster or the cell types, or can color them by the time, and color them by the condition. And then we hover our mouse on the dots. So each dot represents a single cell, and when we hover on each cell, it will show some basic information of this cell. And also we can export this plot to a static one.
Yeah, Tankun, I have a comment regarding PCA here. And we mentioned the PCA several times, which is linear dimension reduction methods. So here you can choose the different factors to show the differences, just to show those factors. And if you can, if you observe those information in the bottom of the figure, like a PC1, 50% variance. And along the y-axis, there is a PCA2, 9% variance. So those shows how strong of the PCA you identified. For example, in this example, the top PC, the first PC can, I could say, can interpret 50% of the phenomenon. Yeah, but the second one can only interpret 9% of the phenomenon. Yeah, so we can see that in this situation, there is one dominant PC identified, and all the others can only contribute a little bit. Yeah, so this is the interpretation of PC. Yeah, and this is why sometimes PCA is not the best choice, because this is the linear one. Yeah. We also provide those nonlinear ones like a Tisney here, yeah, and Tankun, you can show other functionalities, yeah.
Yeah. So I just go through the MDS first. So this is the multi-dimensional scaling. Dimension reduction method is similar to the PCA. It's also a linear dimension reduction method, and we have the similar function here. We can plot them by different factors. And next, for the single cell data analysis, we usually use the Tisney to plot the cell cluster information. And the Tisney is short for T-distributed stochastic label embedding, and it's widely used for single cell analysis. And first, can group the cells by the condition, and the web server we can also plot a three-dimensional make a three-dimensional plot here, and you can easily check these cells are separate between the control and the disease group.
Yeah.
Yeah, next we are at the Heatmap section, and it's also a little bit slower than the usual datasets. So the Heatmap shows top 20 mostly expressed genes in this dataset. So these genes are ranked based on the total gene expression value, and we have you can see the top genes are listed here. And we can also export the data into a static plot here. So you can see in this plot we have the RGS2 gene that are mostly expressed in these datasets. Next, we can do the Byclass analysis of this datasets. So here we can use different algorithms. We provide a list of our Byclass algorithms. First is Cubic, which is a in-house Byclass tool here. And after we click on the analysis, we can check each Byclass that we have identified for these datasets.
I just have a quick question.
Yes, please.
So when we are selecting bi-cluster algorithm and when you just drop down, we see like there are 4 or 5. Is there any preference of using any particular bi-cluster algorithm?
Well, we recommend you just use the cubic. It's
Okay.
it has been recommended by another review paper.
Yeah,
Yeah,
I can follow up with Narin's question. So yeah, cubic is an in-house tool. We do not we do not recommend by ourselves. The recommendation was from a recent review paper published in Nature Communication. They found cubic has the best performance, especially in the single-cell RNA-seq data. So we put this as a default. But I personally believe all those comparisons will have bias, which means we do believe other tools are playing complementary roles in identifying all those biclusters. That's why, besides our own tool, we also implement some other popular ones. We encourage a user to use multiple ones and compare the results, like cross validations, right? So personally, even I'm the developer of Cubic, I know it cannot do everything. Yeah, that's the purpose here, Erin. Yeah.
Okay, thank you.
Yeah.
And another question. So for the cutoff value, so does it relate with the cubic or it's totally depend upon user? It could be like 400, 300, 600, it depending upon the user, or it depends upon or having some relationship with the cubic algorithm.
Naren, the question that depends. Some softwares, they do not have this kind of global optimization methods. They do not have such a parameter. But for some tools like ours, we are more heuristics algorithms, which means we just try to identify a very good seed and then grow the seed into a bicluster. So the algorithm cannot guarantee the global optimization, but it's a greedy algorithm. So in this kind of tools, yeah, not only Cubic, but including others, we need such a cutoff to let the algorithm know when it should stop. Yeah, if you prefer, like there's two kind of situations. If you only want to identify the most significant signals in this data, you do not have to set a very high value, like top 100. Yeah, that's already good enough. Yeah, because of the things that if the bicluster or enriched gene modules, do not appear in top 10 or top 20, you may have trouble in interpreting the results, right? So in that kind of situation, you do not have to set a very high bar. But sometimes your target is not the best one. Sometimes you want to explore what kind of pathways, what kind of the underlying mechanisms encoded, maybe something to be discovered there. So you want to identify more to further for your further analysis. Then in that kind of a situation, I will suggest set a relatively high cutoff to identify more candidates so that you do not have to run it again and again and again. After running one time, you receive the sufficient number of biclusters for the following up analysis. Yeah.
Yeah, thank you so much.
Yeah, thank you for the question. That's a really good question. Yeah, you can challenge every single computational biologist regarding the parameters in their tools.
Yes.
Yeah, thanks for your question. So and this is the results of the bicluster. We are look at the biclass 2. So the bicluster we get we can think them as a subset of block of genes and the cells which they are somewhat similar in this block. So in this biclass 2, so we have 49 genes and 24 cells. And in this block, these genes are somewhat similar, so we can also export these gene IDs and also explore this heat map here. And this is the by-clustering section. So last we have the clustering analysis. So, and there's a warning here that basically means this the clustering method is somewhat very slow here. So, we're running a WGCNA here. This is short for the weighted correlation network analysis, and we also noted that this clustering methods runs best with a large number, that the samples larger than 80. So it's very good for the single cell analysis. So here we have the sample dendrogram and colored by annotated by different colors. And so each row here is the annotation information we just uploaded here. And we have the cell type information, the time information, and the sample condition information. So the similar cells tend to cluster closer. You can see this, for example, in this pink in the condition rows, and these cells tend to cluster together. And we also have the cluster information on gene level, and that's the topological overlap matrix. It combines the gene information here, and can export the file of the PNG and the PDF version here. Can also export the whole gene modules that are co-expressed, predicted by the WGCNA software. Okay, so we are into the discovery-driven analysis. So we are entering the next part of the data analysis. Yeah.
Tankun, let's have a pause here. Since we are transferring from the discovery-driven analysis to the DG analysis, let's see whether the group have any questions regarding those discovery-driven analysis functionalities. So basically, all the functionalities are listed here as correlation, PCA, MDS, t-SNE, Heatmap, Byclass, clustering. So usually with either of them, you can do the discovery-driven analysis. Yeah, they can provide you either gene clusters, gene modules, cell clusters, the features you can train from your data, and the basic correlation things. So usually in your analysis, you may not need to go through every single ones. Like you just want to know whether there is a correlation among your data. For example, you can just do the correlation study on this screen. You can exactly see, there is a very clear correlation patterns among your data, and based on the analysis, you may divide. You may discover a path. Yeah, to further downstream analysis. I'm not saying you have to go through PCA, MDS, t-SNE, and then you analyze all the results. Yeah, you just depends on your question, and then you can select a part of the whole functionality pie. Yeah. Any questions so far? Okay. Tangkun, it seems we are good to go to move to DGE analysis. Yeah.
Yeah. So we are looking at the differential gene expression analysis, short for the DGE analysis. First thing we need to select is the experiment, choose one of the experiment design. So for the default one is the two group comparisons, which are just simply you select two groups in your factor level. Like here we can select two cell types to compare each other. Can simply compare the control group versus disease group under the condition tab. So we also have another other experimental design. So if we want to compare multiple factor, perform multiple factor comparisons, for example, here you can select the cluster information and the condition information. So you can see here we are selecting the control, just these cell types from the control group and this cell type from the control group. So this panel we are combining two factors and perform the differential analysis. And the other one we have other five experimental design tabs. We have the classical interaction design, which you need to select the two factors and choose the reference for each factor. We have the addictive models to select the paired or blocking group. So you need to select the blocking or subject factor and the treatment factor, and also select two reference level for each for the treatment factor.
Yeah. And I have a quick comment regarding the paired functionality here, the experimental design here. So first of all, any questions? Okay. So first of all, I think here should be the most comprehensive differential expression analysis experimental design. Yeah, in the public domain. Yeah, as far as I know. Yeah. So as here you can select what kind of experimental design you want to compare only two of them, and you want to compare multiple factors, consider multiple factors together. Yeah, or like come back to the paired model. So this is feedback from a user. Yeah, we released the initial version without this function, but the user said like in my data there are 5 groups, but I know group 1 and group 2 they are paired. Yeah, this is they should have a different kind of relationship among the other pairs in this 5 group. So in this situation, we build in this model. There is a statistical model. You can have multiple groups, but considering 2 of them are paired. Yeah, 2 of them are specifically correlated. Yeah. So this is additive models for paired or blocking. Like blocking is you want to, when you have one, you want to block another one among the groups. So this is like a co-occurrence or a not. The experimental design is pretty useful when you have this question in your data. Yeah. Okay, Tangkun, that's my quick comment. Yeah, you can go ahead.
And next we have two sections for the main effect experiment design. So you have the main effect group, and you need to choose a main effect reference level and choose the comparison you want to make. And these are the same, similar with the main effects with the group factors. And finally, when this above the experiment design is not in your list, so you can upload a custom design matrix. And so this is required additional information here, but I believe most of the comparisons you will find in the above section here. So I will, for example, I will just perform the very simple control versus disease group. So it will take about 2 minutes. So I will just start submit the function first. So let's look at these DG parameters. So we provided 3 different DGE tools: the DSeq2, the HR, and the Lima. These 3 are very popular, and all of 3 are R-based. So we will be using DSeq2 as a default value, default software, and the other 2 parameters we have here is the adjust p-value cutoff. So the default is 0.05. So every gene that the p-value adjusted p-value less than the 0.05 is considered to be a differentially expressed genes. And we also have the minimal fold change value and the fold change less than 1. If the gene fold change less than 1, this gene will be discarded in the results. So we just finished the differential gene expression analysis. We have the DGE overview. We just selected the control versus the disease. And
another quick comment regarding the DG tools. We currently build in the DSeq2 and IGR and Lima as the options. Lima is very long-term developed existing tools. So we put it here mainly for comparison. Yeah, but we recommend the first 2, DSeq2 and IGR. Both of them have a hypothesis that the RNA-Seq data follows the negative binomial distribution. Based on that distribution, they can identify the differentially expressed genes. Especially DSeq2, they have a new version with new functionality for single-cell RNA-Seq data. So Changkun, I'm not sure whether this DSeq2 is the latest version here, but it's good for you to check the latest version of DSeq. Yeah, they have a version for single-cell RNA-Seq data. Yeah, and HR is another very good example for students who are interested in developing softwares in bioinformatics. HR is a paper published in Bioinformatics. The paper category is so-called application notes. So in such a kind of paper, you do not have to write like a 10-page, 20-page long paper. You can only submit a 2-page like a letter, 2-page manuscript, include 1 figure. And the IGR is even shorter. It's only 1.5 pages, including references. But after this tool released, this paper has at least 7,000 citations in the past 10 years. It's pretty good, example. Such kind of computational technical can attract a lot of attention in the whole domain. Now I think IGR is still a very popular tool. Yeah. Okay, Changkun, show some results.
Yeah. So for the differential gene expressed genes, usually it's split to upregulated genes and downregulated genes. So when we select the control versus disease, so the upregulated genes means the upregulated the differential expressed genes, and the expression value is higher in the first group, which is the control, and in the downregulated group, so which means the genes the gene expression value is lower in the disease, is higher in the disease group, and this is another bar plot. This is showing the number of identified differential genes here. So we have identified about 1,000 upregulated genes and about 500 downregulated genes. So next we are entering the interactive plots, and this is the comparison we just made. And first we have the MA plot, and where the x-axis is represents the log normalized the base mean value, which means the actual gene expression value of that gene, and the y-axis is the log2 fold change. So the fold change is the effect size estimate. So it tells us how much the gene expression same time changed due to the control and in comparison to the disease group. And each dot here is the actual gene. When we hover our mouse on the dot, it will tell us the statistics information like the gene name, the log fold change, and PADJ is the adjusted p-value, and BM is the base mean of that gene. And the plot is connected with this table, so we can click on the table rows, so the plot will automatically highlight this gene ID here. And we can also select multiple genes, and these genes will be highlighted in this MA plot. So this table is the results from the DSeq2R package. We have about 7 columns. First, the gene name, the second base mean, and the average of these normalized gene expression values. And the log2 fold change we just mentioned is the effect size estimate. So since the fold change is the estimate value, so it has a uncertainty associated with it. So we have the LFC-SE, the standard error estimate for the log2 fold change estimate. So we can also express the uncertainty of a particular effect size estimate as the result of statistical test. So it's available in the stat column. So the last 2 columns are the p-value and the adjusted p-value. So DSeq2 performs the each gene a hypothesis test to see whether the evidence is sufficient to decide against the null hypothesis that there's no effect of the. And here we have no effect in the control group on the gene that observe difference between the different disease group. And we usually use the adjusted p-value as the final cutoff, select for the final cutoff. So this is the differential gene expression table. And
Cang Kun,
yeah.
let me quickly jump in regarding the p-value adjustment. Not sure whether this concept is familiar, with to the whole audience. The p-value adjustment is mainly for the false discovery rate. Yeah, which means, give you an example. Yeah, you have a list of gene identified. If you only want to identify the top one, yeah, I mean the most significantly differentially expressed gene, only one. Yeah, no matter what's the p-value there, you should be pretty sure this is the thing should be correct. Yeah, which means the probability you are making a mistake, choosing a wrong one, is very, very low. Yeah, because you choose the best one. Yeah, and then usually in our differential gene expression analysis, we have a cutoff of p-value. We are not select the best one gene. We are selecting a group of genes. So this is so-called multiple hypothesis test problem. Yeah, just think about that. Among all the genes, select the top one. Yeah, making mistake, probability of making a mistake is very, very low. But you select the top 100 gene, then you cannot guarantee every single one in the 100 gene are correct. Are the one, some of them should be false positive, which means when you select 100 gene, the probability of making a mistake for choosing a wrong one is very, very high. That's so-called false discovery rate. And in such a situation, we just adjust our p-value. And the procedure is very, very simple. Yeah, we just make the cutoff more strict. Yeah, for example, using the original p-value cutoff, you can select the top 100 gene as statistical significant ones. But after adjustment, you can only maybe select 56 or 70 something. Yeah. So they will not change the order of the differentially expressed genes. They just give you a more strict p-value cutoff. Then you can only select part of the first 100 genes. So that's basically what p-value adjustment mean. Yeah. And I know some concepts are very common layperson for bioinformatician, but that's always make the biologist confused. Every time you see that, people are talking about the p-value adjustment, but I'm trying to make that layperson enough. Yeah, you know what you are doing for the multiple hypothesis test. Yeah, so this is the comment. Yeah, I'm more than happy to ask questions later today. Yeah, regarding the p-value or something. Yeah, Cangkun, you can go ahead.
Yeah, so we just plot the M and show the MA plot. So it's very common, also very common to have a volcanic plot in the differential gene differential analysis. So the volcanic plot is so the dot, each dot also represents each gene. The difference is that the volcano plot use the log to fold change as the x-axis, and they use the p-value as the y-axis. So we can also highlight this cell here. We can sort this table.
Yeah.
Click on this color.
Similar table. Similar table. Cangkun, you can also select a part of the figures and showcase. Yeah, you can select part of that. Yeah. And the figure will zoom in, for the part you are interested in. And accordingly, the next table, following up table, will update it accordingly. Yeah. Yes.
Yeah. So we're selecting these corner genes, and we just update this table automatically. And these are the differential expressed genes. Yeah. So the IRIS-EDA the analysis ends here. So the next thing we usually do is the functional enrichment analysis. Since we didn't want to reinvent the wheels, we provide this functional enrichment analysis service here. These are very popular ones, like the David, UCSC Genome Browser, and the UniProt. So I can just go to David and about how to use it.
So we are almost through right now.
Yeah.
And maybe we can leave the remaining several functionalities to 3 to 4 sections. And we have 2 or 3 minutes left. Any quick questions before we have the break? Ren, I hope this slide and this link can be shared with the group. Hopefully people can try that during the break. maybe after that we will have some questions. Yeah.
Sure.
Yeah, that's a good idea. So all the students in the program should try this over the next hour, so you can come back with some questions. That the one thing is that taking the time to practice and then coming back and having people right there to help you as you've been working on it will really help you with your coding in the future and your understanding. So thanks, Chin.
Thank you, Rusty, for the comment. And a quick note is for the first time you log in this server to access this server is going to be very slow because this is an RShiny package. For the first time, they need to download, implement every single functions on your computer. Then for the next time, it's going to be pretty quick. Yeah. So if you feel that's very slow for the first time, that's normal. Yeah. Okay. See you soon in S3. Yeah.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.